most important question we wanted to answer is whether you could actually prevent development of specific diseases by having a certain microbiota in early life. But of course, one cannot, answer this question directly without doing causal intervention studies, so that you can study the causal interaction, right? But first, we wanted to see if there's any kind of association. So it's actually prevention more than, direct, implications, you could say. So the design is a prospective study. So what we've been doing is to follow kids longitudinally. Actually, these kids here were born in Sweden in around 2003, 2005, so many years back. And then what we did was to collect fecal samples longitudinally, so 8 time points within the first 5 years. So one at birth, one at within 3 weeks, 2 months, 6 months, 12 months, and then 2 years and 5 years. And then we also had collection of maternal samples, so one pre-delivery and one at 2 months after delivery. And then these kids had blood samples and other samples taken as well. And then we had, in the Nordic countries or in Scandinavia, we have registers. So you get an ID that is personal when you're born. And then one can track all types of, medications and hospitalizations and other types of diseases throughout their life. So we can actually track what these kids were having of diseases later on. Yeah, that's a good question. Actually, it's not really addressed at the moment. So what we've been doing is we identified that they are there, that they are important or seem to be important in early life. So between birth and 6 months of age. So if you have a high abundance of these aromatic lactate-producing bifidobacteria, then they normally also tend to have high production of the aromatic lactates. And if you see this high production, then you see reduced development of food allergies, or at least allergen-specific IgE, so food allergen-specific IgE as a proxy for food allergy, and also atopic dermatitis at 2 years of age. And we even also have data followed up to 14 years of age in these kids that we didn't publish yet, but seeing that hay fever and asthmatic medication was also lower in those kids with the highest abundance of these bacteria, and also the highest levels of these aromatic lactic acids. Yeah, so these are a certain type of bifidobacteria or bacterium that can produce or convert the aromatic amino acids into aromatic lactate or lactic acids. So it's just one enzyme that they hold that other bifidobacteria, they don't have. Yeah, that's a good question also, because actually even though that we see that it can reduce IgE production specifically and not IgG production in human immune cell populations, then we don't know exactly why it is this. We know that humans, but not other, animals, except chimpanzees and so on, they have a specific receptor that can bind to these aromatic lactates, but we haven't been able to see that it mediated via these receptors yet. So it might be other routes for interaction with the immune cells that has just not been discovered yet. But we do see this reduction. So there's no causal link at this moment, as I see it. We have this prospective study where we see associations, but we don't have any, studies that have been designed to actually look at the causal interaction at this moment. But we are undertaking a bigger study in 1,000 Danish infants, where we are trying to study this or investigate this in detail.